Genomic analysis reveals squid and cuttlefish evolved in deep oceans, survived mass extinction in refuges, and later rapidly diversified, following a long-fuse evolutionary pattern. Squid and cuttlefish have long intrigued scientists with abilities like rapid color change and jet propulsion. Researchers have struggled to trace how these features evolved because fossils are scarce and genomic data has been incomplete. That is now changing. A new study published in Nature Ecology and Evolution by the Okinawa Institute of Science and Technology, OIST, combines existing databases with 3 newly sequenced squid genomes. This work identifies the long fuse pattern that led to the modern diversity of squid and cuttlefish, collectively known as decapodiform 10-limbed cephalopod. Squid and cuttlefish are remarkable creatures, yet their evolution has been notoriously difficult to study. The question of their ancestry has been under investigation for decades, and many research groups have proposed different evolutionary hypotheses based on different morphological characteristics and molecular data sets. With this new genomic information, researchers have been able to resolve some of the mysteries surrounding their origins. Squid and cuttlefish live in environments ranging from deep oceans to coastal waters. One feature shared by most species is an internal shell, although its shape varies widely. Cuttlefish have rounded cuttle bones. Many squid species have thin blade-like structures called gladi, and ram's horn squid have spiral shells. Some shallow water species have lost this structure entirely. Earlier attempts to reconstruct their evolutionary relationships were limited by low-resolution datasets that often produced misleading results. Earlier reconstructions of decapodiform evolution were built from datasets with limited resolution and were prone to biased signals, obscuring the true relationships between different species. Whole genome data now provide a cleaner, more consistent picture of how these animals evolved. Studying these genomes is challenging. Squid and cuttlefish genomes can be up to twice the size of the human genome, requiring advanced sequencing technology and significant computing power. Researchers also need fresh DNA, which can be difficult to obtain from species that live in remote or deep-sea environments. Some lineages are only abundant and highly diverse in tropical reef systems like the Ryukyu archipelago, while others are enigmatic and known only in the deep sea. Researchers were fortunate to find some key species on their doorstep in Okinawa and collaborate with colleagues with access to more challenging samples. The study presents the first comprehensive evolutionary tree for decapodiforms based on genomes from nearly all major lineages. By combining genomic data with fossil evidence, the team reconstructed a timeline for how squid and cuttlefish originated and diversified. This analysis shows that these animals originated in the deep ocean, a habitat which still harbors species like the ram's horn squid. The findings indicate that major decapodiform groups diverged rapidly around 100 million years ago, placing their origins in the mid-Cretaceous period. About 66 million years ago, a catastrophic mass extinction event known as the Cretaceous Paleogene (KPG) wiped out roughly 3/4 of plant and animal species on Earth, including the dinosaurs. Researchers proposed that early cephalopods survived by retreating into small, oxygen-rich zones in the deep ocean. The sea surface would have been a very harsh environment for cephalopods. Around that time, very few suitable oxygen-rich habitats would have been found near the shores. Intense ocean acidification in shallower waters would also likely have degraded their shells, so the fact that some form of this feature has been retained throughout their evolutionary history is evidence of their deeper oceanic origins. As ecosystems recovered, coral reefs reformed along coastlines, creating new habitats. Many 10-limbed cephalopods moved into these shallower environments. Following the initial lineage splits in the Cretaceous, we don't see much branching for many tens of millions of years. However, in the KPG recovery period, we suddenly see rapid diversification as species adapt and evolve to new and changing ecosystems. This is an example of a long fuse model, a period of limited change followed by an explosion of diversity. The researchers believe this work provides a strong framework for studying how squid and cuttlefish develop their unusual traits. Squids and cuttlefish have so many unique features compared to other animal groups, making them an endless source of inspiration for scientists. With these genomes and with a clear picture of their evolutionary relationships, we can make meaningful comparisons to uncover the molecular changes associated with major cephalopod innovations, from the emergence of novel organs and dynamic camouflage to the neural complexity that supports their remarkable behavior. Thank you for watching. If you've enjoyed our videos, subscribe to our channel for there's more to come.